as Jim is loading the slides for our next guest, I think that Ingrid herself segued neatly into our next guest, our next presenter, because personalized medicine and targeted therapies really do represent the future of patient-centered care, and that really is the direction we're counting on, I think, our families. And we are thrilled to have as our next presenter Dr. David Goldstein, who's the director of Columbia's Center for Genomic Medicine. And basically, I think it's fair to say they created this for David because they recognize the importance of that genomic medicine and all that it can bring for us. So he's building a program to comprehensively integrate genetics, genomic information, research, patient care, education, all under one roof at Columbia University in New York at the Columbia University Medical Center. Prior to that, he was at Duke University, and if you were here yesterday, and I think most of you were, you heard David Goldstein's name come up from Matt Might at the beginning of yesterday, because of course that's where his journey took him, and David Goldstein was instrumental in helping Matt Might understand what his son Bertrand has and be able to start off on the odyssey from there. Again, we have to start with knowing what we've got before we figure out how we can fix it. And there are a lot of exciting developments happening. Prior to that, he was at the University College of London and Stanford University. So again, I think what you're seeing here is that these experts, it's an international community. They're working not only around the world, but in collaboration and touching various points on the compass. And what we really have is a constellation of experts, and we are thrilled to see so many of them here and working together. Will you please join me in welcoming David Goldstein?
Well, it really is a great pleasure for me to be here. Before I launch into talking about precision medicine and where I see that going, I would just like to make one comment about these kinds of meetings, which are very different from the kinds of meetings that we usually go to. And I would really like to applaud the KCNQ2 Foundation, and I know SCN2A representatives are here as well. I think it's critically important that scientists actually come to meetings like this. One thing to keep in mind is that scientists are people, just like in every other area, and sometimes they need a little nudging to work in the right ways. I think that the competitive spirit in science can be a very good thing, but there are certain areas of work where it's important that there is rather more sharing and rather less competition. And I think those areas include where there is an opportunity to do basic research that can actually identify leads for new treatments. And scientists coming to meetings like this one actually help to remind us why we're doing some of the work that we're doing. So I think it's really important that you continue with these meetings and that you continue bringing us to these meetings and also giving us nudges when you need to give us nudges. So what I'd like to do is try to give you a little bit of a feeling of where precision medicine is going. We are in the very early stages of precision medicine approaches. I want to try to tell you at least what I think of when I think of precision medicine, and also try to give you a feeling of the optimism that at least I feel for where we will eventually get to. But I also want to be realistic that while we're on an exciting path, we actually are on a relatively long path. So what I'd like to do is talk about three types of outcomes that occur when you use genomics to get a clear genomic explanation of disease. I want to say at the outset that I'm actually only talking about outcomes where genomics actually gives us a diagnosis. If you don't find a cause of the disease in the genome, I actually don't get engaged, and so I'm not talking about those examples. But those examples are relevant here, of course, where a cause of the condition has been identified. As Ingrid emphasized, it's critically important that that cause is right. I'm actually not going to talk about that, but I just will emphasize the point that Ingrid made, that sometimes diagnostic labs absolutely do not get it right, and you have to be actually absolutely sure that you have the diagnosis right if you want to talk about precision therapeutics. But there are really three kinds of outcomes that can occur. The first type of outcome is when you get a genomic diagnosis and that tells you exactly what to do therapeutically. And in fact, this is actually the way that a lot of the press talk about precision medicine. It is as if you do something fancy, like a genomic analysis, you find out what's wrong, Then you send your doctor to the right part of the shelf where the medicines are kept. They grab the right medicine, they come back and treat the condition because they know what it is. That actually does happen, and I'm going to give you an example of that. And that, of course, is what we're aiming for. But despite how it's talked about sometimes in the press, that emphatically is not what usually happens. What usually happens is one of the other two examples. The first is the case, so not quite as good as the first one, but at least a direction is set. You get a genomic diagnosis. It does not tell you exactly what medicine to use, but it actually tells you what work to do that might lead to the identification of a new treatment that is targeted. And then the third case is the most difficult of all, where you believe you have a genomic diagnosis, but it really doesn't even tell you exactly what work to do in order to find a new treatment. So what I'm going to do is give you examples of each of these so I can hopefully make clear what I'm talking about, and then, of course, talk about how we can move as many of the examples in 2 and 3 up into category 1. So an example of category 1, which I actually mentioned for those of you that were here last year, from our own work, started with an 18-month-old girl that presented with a very progressive neurological condition that entirely stumped the clinical team. And one illustration of just how rapidly the field is moving is that it really was only a few years ago when we thought, in cases like this, we and others thought that it might be possible to find out what's going on through the use of a very, very new technology, whole exome sequencing, to interrogate the genome as a whole for the cause of disease. This is actually now an entirely standardized approach that has in fact diagnosed many of the patients that are here or represented here. So that's an illustration of how rapidly things are moving. Back then, it wasn't an established approach. The clinical team did not know what the patient had, but they did see that it was so progressive that they feared that she wouldn't live very long, so they did two things. One thing is they decided to treat on the basis of it possibly being an autoimmune condition because they really didn't know what else to do, and it was very progressive. But they also asked us to sequence her and to sequence her parents as quickly as we could to see whether we could see anything in the genome that would A, tell us what she actually has, and B, give an indication of how to treat. When we sequenced her, what we saw immediately is that she has two mutations in this gene indicated at the bottom, SLC52A2, and these mutations really made absolutely clear, really no ambiguity, what the patient actually has. It turns out that that gene was already known to be responsible for a very, very rare genetic condition called Brown-Violetta von Lehr syndrome. And the gene encodes a transporter of a vitamin. The role the transporter plays is indicated in the cartoon. Its role is actually to sit on the surface of cells and to move riboflavin into the interior of cells. And the mutations mean that the protein is not made, and this very rare disease results from not having the protein and therefore not having enough riboflavin inside cells. So what's the treatment for this very rare condition? There's I think 60 patients in the world known at the time that we diagnosed our patient. The treatment is to give patients an awful lot of riboflavin. So this is, in fact, exactly what we're talking about, where the diagnosis tells you exactly what to do and you didn't know what to do before. Now, the reason that this treatment is used is because the canonical way that riboflavin gets into cells is through the activity of this transporter. But as is typically the case in these kinds of situations, there's a variety of less efficient ways that riboflavin gets into cells. And if you simply have enough riboflavin around, then some gets into cells, and then the symptoms of the disease are reduced. Now, the symptoms of the disease include extreme upper body weakness, inability to lift one's arms, inability to hold one's head upright, excessive drooling, eye movement problems, and a variety of other conditions. All of those our patient had, and as I emphasized, the symptoms were highly progressive. One day after the genetic diagnosis, the patient was initiated on treatment with very high dose supplementation with riboflavin. This is a picture, of course, shown with permission of Kara Green comfortably holding up her hands. I can actually personally testify that this is not a staged shot by Kara's parents, because she met the genetics team that actually was involved in diagnosing her. And when she met them, she ran around the room high-fiving every member of the team. I actually continue telling that story because I find that there is absolutely no better way to recruit people into doing genetics research for a living than a story like that. There are a couple, I think, important lessons here. One important lesson is that there really are, when we bumped into this example, we'd only sequenced a couple of hundred patients. So I think there really are, in absolute numbers, a large number of patients like Kara in the world. And this, to me, constitutes an overwhelming argument that we interrogate the genomes of patients when there is a presumed genetic condition that's not resolved quickly, whenever that happens. That is not happening everywhere currently, and I think we all need to be advocates for this to happen. But the other point to emphasize is that as important an example as this is, and to me as uplifting an example about the prospects for precision medicine, we do have to be clear that this is the minority outcome. This is not what usually happens. And we can make that point not only on the basis of experience. The number of times that I've seen this is a very small minority of the times that we've gotten genetic diagnoses, and that's the case for most people. But we can actually see from first principles that this has to be a minority of outcomes. If you think about what's actually happening in situations like this, you start with a patient that you think has a genetic condition, you don't know what it is. You look at their genome and you find that the individual has an already known Mendelian disease. Now, there are something more than 4,000 Mendelian diseases where we know the gene that causes the disease. And amongst those more than 4,000 diseases, it's a tiny handful that are actually treated in the way that this can be treated so effectively. So that means we know this is going to be a minority of the outcomes. And so to me, the focus for precision medicine is to figure out how to take as many of those genetic diseases as possible and move them into this column where we know what to do once we get the genomic diagnosis. So I'm going to now turn to the two other categories of increasing difficulty to try to make the outcomes more like this outcome. The first example, again, those here last year would have heard about this. I'm going to make a couple of new points here and just cover it briefly. But the first example is KCNT1, responsible for now a number of epilepsies, 2 of them indicated here. And the key point is that mutations in this gene have been shown to have a very straightforward functional effect. Work that was done by Steve Petru and others, and I won't walk through this in detail, but what this work has shown is that if you use an extremely standardized assay in the laboratory, where you take the protein that this gene encodes, which is a potassium channel, and you express the mutant form of the potassium channel and the normal form of the potassium channel in frog oocytes, which is a standard way of assessing what these channels do, what you see is that the normal form of the protein, indicated in wild by the WT letters on the left indicating wild type, has a very small current, and all of the mutants, 3 here indicated to the right, have a substantially increased current. Now, there's a consensus in the field that this simple difference between the normal and the wild type forms of the protein directly relates to the way these mutations cause epilepsy. So what I would first ask you to do is just think for a minute about what's happened when you see that a patient has epilepsy because of mutations in KCNT1. You start as a neurologist with a patient with epilepsy. It is fair to say that you know, in fact, nothing, not very little, but you know nothing mechanistically about why that individual has epilepsy. That really is not an overstatement. You then do nothing but sequence the individual's genome, and you find that there is a de novo mutation in KCNT1 that has this property. You now know that that individual has epilepsy because of a particular protein that is generating too much current in some set of cells in the brain. This is actually a remarkable change in your mechanistic understanding of what's happening in the individual patient. And that itself is the starting point for precision medicine. You now have a mechanistic understanding of what's wrong, and you have a direction to go to target your treatment to that precise mechanistic understanding of what's wrong. So that is the case in KCNT1. Because of this idea that there's this simple effect that's responsible for the epilepsy that could be interfered with, Steve Petru himself suggested that quinidine could be used as a targeted treatment in the case of KCNT1 positive epilepsy. And you can see in white the effect of quinidine on reducing the effects of these mutations. On the basis of this work and other works, similar work that has been done, a number of neurologists have initiated patients of theirs that were judged to have KCNT1 positive epilepsy on treatment with quinidine as a targeted treatment for the epilepsy. That was done first by Dennis DeLugos, you're going to hear from later, and I believe, and his colleagues at the Children's Hospital of Philadelphia, and it was done later by colleagues of mine at Duke. The first experience actually was somewhat encouraging. The patient achieved seizure freedom for the first time in some time. The experience at Duke was less encouraging. One of the patients had their seizures apparently reduced somewhat. The other patient had their seizures not changed very much. I want to make a couple of points about this. Before I do, let me just make a disclaimer. As I think many people here know, I am not a neurologist. I'm not a clinician at all. So I want that to be understood. But I'm still going to share my own personal impression because I've talked to a number of colleagues that are neurologists that have treated their patients with quinidine. And I will share my own personal impression that quinidine is actually doing very little or perhaps nothing therapeutically. So I don't think that quinidine is going to be the way that we treat. I think that that's actually not very surprising in retrospect. A key reason it's not surprising, as you can see in this figure, that quinidine only modestly undoes the effects of the mutations. And on top of that, quinidine is actually toxic for the heart in high enough doses. And so I think fundamentally we're limited in not being able to give patients enough quinidine to have therapeutic effect without creating intolerable cardiotoxicities. However, this example does illustrate how easy it is to go from the identification of a targeted treatment to its evaluation in an off-label setting. And what that means is that when we find new, more selective, therefore safer inhibitors of KCNT1, we will be able to quickly evaluate them in patients when they are already approved drugs. And of course, they can be explored in the context of new drug development. So I do think that this is actually very encouraging. And I personally have, in fact, a very high degree of confidence that we will be treating KCNT1 at some point with a targeted therapy. The other comment I want to make about this, and you learn as you go, the initial impression of quinidine in the community was that it might be helping some. And my own view, and not all of my colleagues may agree with this, so it really is my own view. My own view is that when we first test a targeted medicine, as off-label, for example, I think we can more or less count on the impression of a positive response. And this is actually something I think we have to keep in mind and be very, very careful about in the context of bringing out targeted treatments. The reason for that is that we don't use them in a blinded fashion when they're evaluated in this way. And everyone carries expectations. And that is not just the patient families. I would emphasize that the neurologists and the scientific community, me included, carry expectations too. We are all excited about targeted treatments. We all believe that's the future. And when we first start trying a targeted treatment, we're going to be looking to see that it actually worked. And so we have to be very, very careful about what we suggest, knowing that because of expectations, when they are first evaluated, either the clinician's expectation or the family's expectation, there will often be an impression of a positive response. This suggests to me that it's important that we be as careful as possible in the research community in making suggestions about targeted therapies, because we really want to be careful what we put out there, knowing they probably are going to be tried for serious conditions, and that at least initially there's going to be an impression of a positive response. Now, the lesson from that for me is that if suggestions are made about targeted treatments for repurposing opportunities, that means taking an existing medicine and using it where you otherwise wouldn't have, like quinidine, I feel that if anybody is going to make that suggestion, they have a responsibility to evaluate comprehensively what the options are for repurposing. We didn't do that. I'm not criticizing the work. I was involved in it. We didn't do that in quinidine because this is just starting. But it can be done. That assay system that I mentioned of putting the proteins in the oocyte could easily be used to screen, for example, all approved drugs that cross the brain-blood barrier. So if it's the case that right now, today, there's an already approved drug that is safer and works better than quinidine, we would have known about it had that screen been done. So I am suggesting, again, speaking for myself, not my colleagues, that whenever we're going to make these suggestions, knowing what's going to happen downstream, that we should be as comprehensive as we possibly can be. And I actually would like to see us work in that framework for KCNQ2 and in other genetic epilepsies. The third category of outcome is when you get a clear diagnosis, but it actually doesn't give you any clear starting point for running screens that can give you a targeted treatment. You heard yesterday about an example of that we bumped into and that you can read more about in the New Yorker. We sequenced Bertrand Might, saw that he carries 2 mutations in this gene, NGLY1, which is now known to cause a syndrome called NGLY1 deficiency. I want to make one comment about the process that led to there being a story in the New Yorker. And the comment is that when we sequenced Bertrand and his parents and saw that he had mutations in NGLY1, that gene actually hadn't been implicated in a human disease before. The syndrome that eventually was described wasn't known. And so when we looked very carefully at Bertrand's genome and saw these mutations, we thought for a variety of reasons that the mutations in this gene were likely to be responsible, but we couldn't be sure because it was just one patient. But we actually decided, which was counterculture at the time, we in fact required permission from the dean to do this, we actually decided that we were going to communicate our suspicion to Bertrand's parents that this is what was responsible. Not our certainty, but our suspicion. A number of reasons that we decided to do that, but one of them was we felt that his parents would understand that we suspected, but we're not certain. So we did that. We communicated the results. We thought that this was a new syndrome due to these mutations in NGLY1. And his father, who you heard from yesterday, I remember this very clearly, told me, okay, so let me see if I've got this right. You think it's mutations in NGLY1, but you are not going to know for sure until you identify more patients that are like Bertrand and that have the same kind of mutations in NGLY1. And I said, yep, that's exactly right. He said, okay, great. We'll go off and find additional patients that confirm that this is right so that more work can be done. And I remember thinking and did not say, great, you go ahead and do that, and we'll also look for more patients and we'll find them, and then once we do that, we'll let you know that we confirmed the diagnosis. You will now know from yesterday that over the course of the next however many months, Matt Might, through his blogging, and his wife, were directly responsible for the identification of an additional 8 or 9 families that definitively confirmed the diagnosis. One of the ways this happened is that Bertrand has some very specific features. We may or may not have found one additional family on our own. The confirmation happened dramatically faster than it would have happened without their involvement. And this, to me, provides really an overwhelming argument for the inclusion of families as partners in the research, in particular, for rare conditions. And in fact, that's, of course, the reason that there is a New Yorker story, and I believe that this has really helped to make the case that families ought to be engaged in the research and has actually helped to move us away from some of the paternalism that had characterized some of the ways that this work had been done. Unfortunately, however, knowledge that it's mutations in NGLY1 that are responsible does not, in fact, allow us to run a screen like we can in the case of KCNT1. We actually don't have any assay in the laboratory that can show an effect of mutations in this protein that we know is related to the way the mutations cause disease in patients. So we actually know this is responsible, but we don't know what to do next. You can start exploring, but we don't know what to do next. This is the most common outcome. And if we actually want to systematically approach precision medicine in these genetic conditions, we need to figure out a way to take this as a starting point where the mutations do something, we don't know what it is that they do that's relevant to how they cause disease, and be able to use that as a starting point for screening. There are many, many other examples like that. I'll highlight just one of them from work that we've been involved in as part of the Epi4K collaboration, also involving another collaborative group being based in Europe, Euroepinomics. There are many representatives of Euroepinomics here. We implicated Dynamin-1 as a new epileptic encephalopathy gene. Some work done by Ryan DeHinsa in my lab and others have shown that these mutations have an effect on a biological process called endocytosis, which is involved in synaptic vesicle trafficking, a key way that neurons communicate with one another chemically, a key part of the process of chemical communication for neurons. And the point to emphasize here is that while we are able to see that these mutations have this effect on synaptic vesicles, we don't actually have an immediately available screening approach that we can use. And I put this up in particular just to emphasize that the very simple approaches like expression in a frog oocyte obviously can't be used here because these mutations appear to actually screen on. We need an experimental platform that has neurons communicating, which is not sticking a protein in a frog oocyte. So I will just say, so it doesn't all sound like doom and gloom, I do feel that there are directions that are being followed now that have the hope of establishing for us these more general screening paradigms that will be able to reflect in principle, the effects of lots and lots of different kinds of mutations. What we've been pushing very aggressively is the idea of using cultured neuronal networks. Here what you do is you take hundreds of thousands of neurons that have been engineered to carry the relevant mutations, and then you let them actually form a network in the laboratory, and then you listen in on the way those neurons fire in the network. And you try to see whether the mutation has an effect on network behavior. So the way this is done is you take these microtiter plates, you seed the wells with neurons that do and don't have your mutation. The plates have electrodes embedded in them, and then the electrodes actually record the firing of nearby neurons. And what we're looking for are networks that behave differently with the mutation in comparison without. Now, when I started getting interested in doing this some years ago, there was actually a lot of skepticism that we could reveal relevant characteristics in these networks that we could then use for screening. And so what we cast around for was an illustration project that would convince the skeptics that this could work. And what we finally settled on, sorry about the slide, what we finally settled on is taking a particular genetic perturbation, which is related to a particular microRNA. But the key point is that if you take this particular little RNA out of mice, they develop a profound seizure phenotype and die early. So it's a very simple genetic perturbation that has a very profound phenotype in the mice. So of course what we wanted to know is, well, do we actually see some version of that in these cultured neuronal networks? Because if we do, then the idea would be that genetic perturbations that are relevant in mice and genetic perturbations that are relevant in humans, causing, for example, human epilepsies, should be viewable in some fashion or recapitulated in some fashion in these cultured neuronal networks. So this is just an illustration that taking this microRNA out of mice has a profound effect. And what we then did, of course, is take it out of these cultures. And I won't walk you through these slides, but what I'll emphasize is that what we found when we removed this microRNA from these neuronal networks is that they became more active immediately. That's what's shown on the left-hand side. They immediately became more active. So this perturbation, genetic perturbation in the mouse that causes a seizure phenotype in the mouse, has a clear recapitulation in these cultured neuronal networks. Now, what's the importance of that? The importance there is that what I can now do is put compounds into the wells there and try to push the mutant behavior shown there in red back towards wild type. And I will emphasize that we are now pursuing this for a variety of different genetic epilepsies. And we have shown that in a number of cases, we do get clear phenotypes. So I think we really do see here the beginnings of what can be viewed as a general paradigm for modeling these mutations. Now, one would not suggest that there's anything like a realistic set of neuronal networks represented in these cultured neuronal networks. But the key point is, in these cultured neuronal networks, neurons communicate. So the machinery that neurons use to communicate is represented there. So we can still reveal effects of the mutations. Okay, so I don't know where that slide's back up. So what this really outlines for us is what you can consider a truly novel paradigm for drug development. And this novel paradigm is to develop appropriate models that can be screened, you can actually test compounds on, based on causal mutations, based on the mutations that cause disease. And I will strongly emphasize that this is an entirely new way for developing, almost entirely new way for developing treatments. Vast majority of drugs that are out there have not been developed on the basis of models based on the mutations that are responsible for disease. This is a different way of developing treatments. Once you have these models, you then screen on the models to find compounds that either directly undo the effects of the mutation. So for example, in the KCNT1 example, you have too much activity, you find an inhibitor that acts directly against that activity. Or critically, you find a compound that compensates for the effects of the mutation. Now this is where it's so important that you have complexity in your model. If you're actually going to take the oocyte as a starting point and screen on the effects of the mutant on the protein in the oocyte model, the only thing you can find is something that acts right on that protein, because there's nothing to compensate there. But if you use a cultured neuronal network, for example, or of course an animal model, but a cultured neuronal network, there are lots and lots and lots of other things that neurons do that are represented there. So you actually have the hope of finding a compensatory mechanism. So once you've done your screening, you then take, of course, the results that come out of that, and you test them in individuals that have disease because of those relevant mutations, so in the appropriate genetic subgroup. But once you've done that, you then move on and ask, does it work in anyone else? And there are actually reasons to believe that compounds developed for genetic subgroups of diseases will actually have application in other patients. One way to illustrate that is that, for example, in epilepsy, most anti-epileptic drugs actually include sodium channel blockade as one of the ways that they work, and it's a minority of patients that have epilepsy because of identifiable mutations in sodium channels. I also want to make one quick comment, a little bit less relevant to this group, but one of the criticisms actually of a precision medicine approach in epilepsy and other diseases is that it will only apply to a very, very small proportion of patients. Well, I have one comment first off, which is relevant here. Even if that's true, it still matters for those few patients where you actually do develop with a precision medicine approach. But the other comment is, I actually think that the application will be broader than a lot of critics of precision medicine think. And our illustration of this comes again from the Epi4K consortium. I won't go through the statistical genetics represented here, but what this slide illustrates is that one of the most common forms of epilepsy, what really would be considered a garden variety form of epilepsy, non-acquired focal epilepsy, is influenced by all of these genes indicated in blue, and all of those genes indicated in blue are also responsible for rarer, familial, and more devastating epilepsies, showing a very clear overlap in the genetic control between rarer, more serious epilepsies and more common epilepsies. So that's it for the science. I would like to just conclude with a few words about the approach that we're taking to precision medicine at Columbia. Very happy to say that Columbia decided to create a truly integrated program in precision medicine that is in fact university-wide, not just the medical school, but all the way across the university. And one of the reasons for that is the recognition that precision medicine isn't only about the genomics. That is actually how it's described sometimes, but as I hope I made clear in talking about the science, it really is about using the genomics as a starting point to first get into the biology of disease, and then secondarily use that insight into the biology of disease to develop targeted treatments. And we developed a paradigm that allows us to pull together the investigators that can take this kind of an integrated approach. Within the Institute, we have pulled together a variety of basic infrastructure that's needed to allow us to not only try to diagnose patients genomically and get that right, but then also use the results as starting points for modeling, both in mice and in cellular systems like I described. And yesterday, some of you may have talked to Wayne Frankel, who's in fact running the mouse modeling programs within the IGM and in fact for the overall Columbia Precision Medicine Initiative. There's a variety of modeling paradigms that we're pursuing. I won't walk through them, but instead what I'll do is actually illustrate specifically what's happened so far over the last year in one of the big initiatives that's run out of the Institute, which is of course focused on epilepsy, a primary interest of mine. So over the last year, we have been offering whole exome sequencing to every, independent of ability to pay. Doesn't matter, in fact, we don't charge any of them anything, or their insurers right now. So independent of any ability to pay, any patient that comes through our health system that has an unexplained epilepsy, so there's no obvious explanation for the epilepsy, is offered participation in a genomics research study. The vast majority of them that are offered actually agree. So we have now, through this effort, sequenced 251 patients with unexplained epilepsies, 80 of them in a so-called trio design, where we sequence the patient and their parents, and the rest of them as singletons. And out of all of those, we have 14 genomic diagnoses. The genomic diagnoses are indicated there on the right, but this illustrates that there are diagnoses that you will get from doing this kind of genomics that would otherwise not be in the medical record. These individuals are not otherwise being sequenced. So what this means is that as targeted treatments become available, candidate targeted treatments, we can go to these patients' medical records now and tell them that there is a targeted treatment. Do you want to test it? Because we know that this is the explanation. So I really believe that this is what we need to do. And in fact, we, having now identified some of these in our own patients, have programs running to develop those targeted treatments for a number of these conditions. The final comment I'll make is that there, of course, now is tremendous momentum in precision medicine, in part because of President Obama's precision medicine cohort program. We are, in fact, a part of that cohort program. The initiative aims to enroll 1 million people in a cohort study that will eventually include genomics. Our part of that is to enroll 150,000 people from our own health systems in a partnership in New York that involves Columbia, Weill Cornell, and also Harlem Hospital. And this really just is meant to illustrate what kind of a scale we're actually moving into in precision medicine. And these kinds of approaches actually will make us better and better and better at the genomic side of precision medicine. So much so, in fact, that I will admit that I actually am moving progressively towards ignoring the genomic side, which is how I made my career, and emphasizing all my attention on what happens next. That's where we really need to work hard. So with that, I'll end, and thank you very much for your attention. Please.
Thanks for coming in. Last year there was a question asked about inhibiting the sodium to get a potassium
I'm not sure. So I mean, I guess Ed, I think, would be in a better position to comment on that. But I guess what I would emphasize is that it's very hard to predict what's going to happen. Is basically the experience. I mean, we have these extremely simple experimental systems, and we look at particular effects in these simple experimental systems. We don't actually know what's going to happen until we get in really realistically into animal models. This is why it's actually of absolute critical importance that we develop and fully characterize a number of knock-in models for KCNQ2. This is the overwhelming priority right now, because if we actually have good models, then it's reasonable to assume that what happens in response to treatment in those models will be somewhat predictive, at least what happens in people. Should it be a priority if it hasn't happened? If you already have a diagnosis, or if you don't? It's not. If you have a diagnosis and it's right, then I don't think there's any need to have further genetics done. Our capacity to recognize things that modify the presentation is basically nil. We just basically can't do it. So I don't think it adds value if you have the diagnosis right. But you need to have the diagnosis right. Sometimes what happens is there's a possible diagnosis, but then you look at the rest of the data and there's a better candidate somewhere else, and so that's a reason to have the rest of the data. But if there's confidence of the diagnosis, if it's a highly recurrent site, or there's other reasons to be confident, then I don't see any reason to do the sequencing. Please.
I didn't happen to see the KCNQ2 on the slide, one of your last slides, sort of like one question. Then second, where would you put us in this room, as it pertains to those three different levels that you outlined initially?
I was afraid that I was going to be asked that. It's a great question, and so first let me the easy one. So we're actually not currently in the Institute doing much work on KCNQ2. I think that's becoming increasingly likely to change. That I think we're probably going to get involved, at least with cultured neuronal networks and maybe in some other ways. As far as where to place it, it clearly is not one. There's no question about that, because there isn't a highly effective treatment, even if there are openers that work a little. It's clearly not one. It's clearly somewhere between the second and third category. And I say somewhere between because it's not good enough to get a functional effect of a mutation. You have to have a functional effect of a mutation that you believe is related to the way the mutation actually causes disease. That's where it's difficult. And so that's why I was asking Sarah the question about, for the putative gain of function, what really is happening? Is it an increased current that is responsible for disease in those individuals, or is it something else? And it is often extremely difficult to know. And so I would say it's really somewhere between 2 and 3.
I've asked this question in the past, but it's been a couple of years. Gene therapy, is that a precision medicine?
Gene therapy is most certainly a precision medicine approach. I'm personally not paying too much attention to gene therapy because I'm interested in brain diseases, and I am not, in fact, optimistic about the application of gene therapy approaches in brain diseases in the sufficiently near future. I'm just not very, there are a lot of challenges about delivery and safety that are soluble challenges. They will eventually be worked out, but I actually think it will take a long time to work them out, and so I'm more focused on small molecule approaches and other approaches than gene therapy. I can also say that it's partially a career choice. I mean, I really do believe that it will take time. I'm 52, and I'm too impatient. Now, if you're doing other things, not brain diseases, that is a way to go. But I really think it'll take a long time. It's my own view. But there may be reasons to be optimistic that I don't know about. Yeah, please.
Do you think it's likely that in the future, a cultured
Absolutely. I mean, there's that. So so I think that's to me a priority. We want to know whether we get relevant behaviors in those networks, and then if we do, we want to screen on that. So that's exactly right. But I think that is I really believe is a general approach. So the first important question is whether there is a clear behavior of the network, what we call a phenotype.
It's exciting to hear that you think KCNQ2 might be something that you all work on. What does it take to get that to happen?
Oh, we just have to think, and just find time to think. But I think Wayne and I have started discussions. I think eventually that will happen. Please.
So is a way to get our name KCNQ2 on that list to register for precision medicine,
For the institute, no. I think that we're going to try to look at the network, so I can make that commitment right now. So it's. I mean, there are a variety of things that draw us into it, but one of the things that draw us in is that there's we hope for doing something useful ultimately. That's one of the primary drivers of what we do. And so the hope would be that, if we find something, that there is a community ready to try to figure out appropriate ways to make use of the findings, and that certainly is the case for KCNQ2. So that constitutes a motivation. KCNQ2 also, I think there are reasons to think that there ought to be a relevant phenotype in these cultured neural networks. So my guess is we'll actually see something. So we'll see. So we will eventually look at that one way or another.